Muy buenos días a todos. Me da un gran gusto darles la bienvenida a esta sesión de la serie Fronteras en Sistemática, Biodiversidad y Evolución del Instituto de Biología. En esta ocasión es con gran gusto que tendremos una charla por el doctor Timothy James de la Universidad de Michigan llamada Exploration of Fungal Diversity Using a Single Cell Sequencing Approach. A continuación, con mucho gusto, le voy a ceder la palabra a la doctora María del Carmen González Villaseñor, quien nos presentará al ponente. Muchas gracias, muchos saludos a todos y adelante, Mari Carmen.
Muy buenos días a todos. Estoy aquí para presentar a nuestro distinguido conferencista del día de hoy, el doctor Timothy James. Él es profesor de Ecología y Biología Evolutiva en la Universidad de Michigan, en Estados Unidos. Es el curador de la colección de hongos del herbario de la universidad y ocupa la posición honoraria instituida en honor al valioso legado de los doctores Lewis E. Wellmeyer y Elaine Prime Wellmeyer para el estudio de la taxonomía de los hongos. Él recibió su licenciatura en Ciencias en Botánica en la Universidad de Georgia, en Athens, Georgia, en Estados Unidos, y su doctorado en Filosofía en la Universidad de Duke, bajo la asesoría del prestigiado doctor Richard B. Gates, en Durham, Carolina del Norte, Estados Unidos. Sus estudios se enfocaron en la reconstrucción del árbol de la vida de los hongos y también aplicó la genómica para investigar los sistemas de apareamiento de los hongos, su ecología e historias de vida. Sus trabajos sobre taxonomía de los hongos incluyen los hongos zoospóricos y otros linajes con divergencia temprana, particularmente los que no se pueden cultivar en el laboratorio. Actualmente está enfocado en el desarrollo de métodos de genómica unicelular para la exploración de la diversidad fúngica, la relación entre el genotipo y fenotipo, los micovirus y la función de la recombinación mitótica durante los ciclos clonales. Ha participado como concejal de la Sociedad Americana de Micología, conocida como Mycological Society of America, y en varios comités de la misma sociedad como juez para otorgar los premios a la investigación, premios a los mejores estudiantes, distinciones académicas y también para organizar la conferencia anual Carling, en la que invitan a un micólogo sobresaliente y consolidado en micología. Él es coeditor del libro titulado El reino de los hongos, The Kingdom Fungi, editado por la prensa de la Sociedad Americana de Microbiología. Y también es editor asociado de la revista Mycologia, que edita la Sociedad Americana de Micología, que como ustedes saben es una de las revistas más prestigiosas en el ámbito internacional. Otras de sus funciones son como director del departamento de Frontier Master Breeding Program. Es cofundador y director de la Información Micológica Americana del Medio Oeste. Es una asociación educativa sin fines de lucro que en inglés se conoce como Midwest American Mycological Information, MAMI, que una de sus principales funciones es en la retribución social, como por ejemplo educar al público y también mejorar la comunicación del conocimiento y tecnologías entre el público, el gobierno y la industria. Es miembro del Comité Ejecutivo de Usuarios del Department of Energy Young Genome Institute. Tim es también miembro de la Sociedad Americana de Microbiología y, sobre todo, su cargo más importante es que es un miembro del Instituto Canadiense de Investigación Avanzada, el reino de los hongos, amenazas, oportunidades. Y bueno, sin más, le vamos a ceder la palabra al doctor James para que nos exponga su interesante plática.
Thank you. Should I start my slide share now?
Yes, sure, Tim. You can go ahead.
Great. Thank you so much for the invitation, Susanna, and thank you, Dr. Carmen, for the wonderful introduction. I'm really honored and excited to be here. It's a great opportunity to speak to you about my passion for the fungal tree of life. And I'm really pleased to see Systematics being so well represented in terms of the science that's going on in your institute. I have a long-standing goal for understanding the fungal tree of life, its shape, and all the diversity of the many branches. I'm particularly intrigued by lineages that are difficult to work with, because these are ones that produce so many insightful surprises when you dig down and try to see what their underlying biology is. The sort of centerpiece of what I'll talk about today is our work using single-cell genomics to understand the fungal tree. The tree of life is full of many diverse lineages, and we are only privileged to see the tips of that tree. And, it sort of looks like this great diversity and history and interconnection, but we're limited to observations of the tips. And those tips present characters, like a green and a red character state. And they also, are united by an evolutionary history, the underlying tree structure. And so the idea is that, we only see parts of the tree that we know about, and we can only infer relationships among the taxa that are sampled. But there's all this dark area of the tree, and in some of those are lineages that are poorly described, and for various reasons we might be sampling. We have biased sampling and don't know those lineages very well. But if we did know those lineages, we would see a much richer and fuller part of the tree. And for example, in this example where we have the green and the red characters, we might see that there are green lineages that go deeper into the tree that we just didn't know about. We never sampled them. And in adding those to the tree, that completely changes our understanding of the reconstruction of this green-red character state. So it's really vital for us in terms of understanding the evolution that's occurring along the tree to be able to sample all of the lineages that we possibly can. And some examples that you might find in fungal biology of characters where additional sampling can reveal a much different character history would be things like a flagellum. So most fungi don't have a flagellum, but as it turns out, most of the early branches on the fungal tree are groups that either do or did at one time have a flagellum, and that character has been lost multiple times in the history of fungi. And another character might be the use of this 21st amino acid selenocysteine. It's a rare characteristic, but it's one that's shared by many eukaryotes. And it turns out that early branches on the fungal tree have used this amino acid and still use it. And it is only more recently that that most of the fungi have stopped using it. And knowing this about the history of this group of organisms like the fungi and these traits gives us an idea of what was going on in the past. The fact that most of these early branches are flagellated tells us that the habitats that they were living in and their modes of nutrition could be based around an aquatic habitat. Now, as a little bit of a backdrop, we're going to talk about the many ups and downs of fungal taxonomy, starting with sort of the first phylogenetic tree of organisms, which put fungi in kingdom Plantae as a tiny little twig related to the lichens. And it was sort of this old-fashioned concept that we're moving away from, and an appreciation that actually fungi are not plants at all. Instead, they're more related to animals. So this is a really fun figure which shows animal diversity here. And then, we have fungi as related to the animals now instead of plants. Okay, so that's an improvement. But the amount of diversity in fungi is not appreciated, at least in this particular figure. But we actually know that there are many more fungi out there. At least that's what the consensus is among mycologists, is that there's a great diversity of species. And I'm going to call that diversity dark matter because we don't have a name for it. And in a lot of cases, we don't even know its underlying biology. So quite a long time ago, we estimated that the total diversity of fungi is roughly 1.5 million species. Nowadays, that's considered a low range on the estimate of total species diversity. Currently, we're at roughly 150,000 species that are described. So you see that the described diversity is much lower than what we perceive to be the total diversity. Now, we rely heavily in mycology on sequencing of species in order to determine when we have new species. And there are only about, a third of these species that are present in GenBank. And probably the most important metagenomic study to date was a massive global sequencing program that looked at soils from throughout the world and tried to characterize the diversity in those soils, and recovered an approximately 45,000 species using these molecular barcodes, of which those only about 10% overlapped with stuff that was in GenBank. Okay, and I will contrast that a little bit. So, for example, if you were to just go out into our backyards and culture from soil, for example, we would only recover maybe 100 species, which is a very small proportion of that species diversity. So, in essence, what I'm arguing is that most of this part of this fungal pie is largely unknown, and it is this dark matter. And we, as a mycology community, need to better understand these organisms that are in this part of the pie. So for the rest of the talk, I'm going to give a little bit of an example of why we need to go beyond metagenomics to try to essentially put morphology and put function to some of these sequences we know exist. On the way, we will describe the systematics of the chytrid fungi, the aquatic flagellated fungi, and why we're focused on them as a lineage of particular interest for our part 3 single cell approaches, where we are trying to link morphology and function of these dark lineages, the stuff that we know is out there based on environmental DNA sequencing, but we don't know what it does. All of this is done in a phylogenetic context. Okay, now part 1 of this talk, we're going to discuss how everything now is reliant on sequence DNA sequencing, and how that's both, a good thing and also a limitation to our understanding. High throughput sequencing really changed how we look at fungal ecology and fungal communities, and it really opened up the gates for discovery of new species. And that has been a real blessing, because we now are able to actually know what's really there, as opposed to relying on fruiting bodies or relying on cultures. we can actually get a better estimate of the fungi that are present in the environment. How this works is familiar to all of you, but basically we as a mycology community are focused on small sequencing regions that are targeted by next-gen methods like MySeq. And we will sequence small, highly variable regions like the ITS2 region or maybe the ITS1 region. These are great regions for barcoding because they evolve so quickly and have little constraint on their sequence evolution that they're usually, almost always, diagnostic for a species. So we use them as barcodes for species in communities. And this is how a lot of the discovery of this dark matter, fungal dark matter, has played out. Now, that's got one limitation, which is that you have a very highly variable gene region, but that makes it difficult and in most cases impossible to make a phylogeny of all of the stuff in your meta barcoded community. So if we sequence soil with ITS marker, we can kind of make these species numbers, and if we're lucky, we can use BLAST to try to say what that molecular signature is from, like taxonomically speaking, but in some cases we can't really get to a high resolution. And for example, in this classic study that I mentioned, 6% of the sequences could not be assigned to a phylum, which really tells you, means we really know nothing about it. And so this is work that was done by Lehu Tietersu's lab, and he was the one who led that global metabarcoding study. But given that there were these OTUs that couldn't be assigned to phyla, how could you figure out what they are? Well, if you could put them in a tree, you could likely do the work. And so what he did was he designed species-specific forward or reverse primers to try to sequence out into the more conserved regions, and then made a tree of all the things that could be treated in this way. And all the red in this tree are the things that were previously not assignable to phylum, but now could be placed. And you see lots of interesting things here. Some things go in groups we're quite familiar with. So here's the mushrooms, the agaricomycetes, and we see some interesting diversity in parts of that tree. We also see some ascomycetes. A lot of them are in long branches. And then we see things like many sequences that are in the Rosellomycota, also known as the cryptomycota. And these are mostly intracellular pathogens that have a very cryptic biology and are in hosts like amoebae and zooplankton and water molds. And so they've been largely overlooked by biologists, but are extremely common in soils and in aquatic habitats. This other group that's being highlighted is a clade that's related to the archaeorhizomycetes. And I'm going to now talk about the discovery of archaeorhizomycetes as sort of a foundational example of how we need to go beyond just sequence information. And how we can do that is by culturing. But the archaeorhizomycetes started out as something called soil clone group 1. Okay, and as soon as we had started sequencing soil, it became apparent that there was this one group, the soil clone group 1, that was really common and found in pretty much every study. But it was so phylogenetically distant based on ITS and LSU that we really didn't know what it belonged to. And what happened was that in Uppsala, Sweden, there was this long-term program of culturing fungi from plant roots. And they had tens of thousands of cultures and were kind of working their way through barcoding all of these fungal cultures. And actually had finally connected the dots between a sequence that they had generated from one of these cultures and this soil clone group 1. And so I guess the point I'm trying to make is that all of a sudden they had this culture, and now they could start to ask questions about what this thing could possibly do in nature. And what they showed was that you could actually infect pine roots, and that it would grow on a pine seedling, and would grow on the outside of it, but not make any kind of mycorrhizal type structure. So the symbiotic relationship is still unclear, but they were able to at least, begin to manipulate the culture, look at its morphology, see how it grew in culture and on the host, and test for metabolic usage of carbon and nitrogen. So these are kind of painstaking studies, and it's very hard to connect the dots between environmental DNA sequences and cultures. So we need another approach. And lastly, I just wanted to show where this thing went in the tree. It goes into this oddball group, Tephrinomycotina, and these are the things that it's related to. And as you can see, morphologically, they're very different. So now I'm going to warm up the single cell discussion and talk about one of my passions, which is the systematics of zoosporic fungi. And the zoosporic fungi and the whole kingdom go back quite deep in time. So we generally think of animals and fungi, splitting from each other, roughly 1 to 1.5 million years ago. And then around 1 million years ago, we start to see the diversification. Sorry, not 1 million. 1 billion years ago, we start to see the diversification of fungal lineages. And all of these basal lineages are things that most people don't think about and haven't studied, although they do include important fungi like the Glomeromycotina, the pin molds, and then the zoosporic fungi that I study. And up here at the crown here, this is the Dicaria. So all the cup fungi, the yeasts, and the mushrooms. So one of the point I wanted to show is that a lot of these lineages had appeared deep in time, and that by the time you get to the Devonian, we start to see a diversity of things. These are zoosporic fungi, and we see various chytrid-like structures, which chytrids are kind of like, it's a word that's Greek meaning little pot, and this is a sporangium in a fossilized tissue. And you can see here the rhizoids coming from that sporangium. This would, I guess, be probably some plant material. And then this is a fossilized Allomyces, a water mold, and it's incredibly similar to extant Allomyces. So it's very easy to place this onto a tree. Okay, so these are examples of zoosporic fungi from the Devonian, and it is a time that was quite warm and quite wet for our planet, and already full of land plants. Pictured here, lycophytes. And so those habitats, are different, but in some ways similar to modern day habitats. And it is those kind of habitats that we see chytrids in today. So this is a fen in northern Michigan, and it's one of the sites where many of my predecessors, such as Frederick Sparrow, studied and discovered many chytrids, and where we can still go today and find a huge diversity of chytrids. So I'm using this term chytrids, and in this talk, I'm kind of using it for all the fungi that have swimming spores. Okay, that sometimes you'll hear the term chytrid, and it's really kind of restricted to the fungus that is a chytrid that is pathogenic to amphibians. And here's my colleague Joyce Longcore receiving the Golden Goose Award for her contributions to the discovery and description of this chytrid that's pathogenic to amphibians. That is not like any normal like chytrid, actually. So of all the maybe 1,500 described other chytrid species, most of them don't attack vertebrates. This is in fact the only one that's known to attack vertebrates. And they look like this, sort of, here's the sporangium and here's their rhizoids, here's their swimming spores. And this is showing some sporangia at maturity. There's a great diversity of named phyla that are zoosporic. So we have 1, 2, 3, 4, 5, 6, 7, 8 phyla of zoosporic fungi. The ones in green were emphasized as being where the primary algal parasites are, and that's one of the functions they have in ecosystems. The other function they have is as a degrader, and they can easily be recovered by culturing techniques. If you see spring pollen blooms that land in ponds and lakes, those will often be colonized with chytrids, like in this picture. And they like to go after recalcitrant substrates, like also including hair and skin, like snake skin, cellulose. So they're easy to recover with baits. But as I mentioned, there are these algal parasitic chytrids, and those are a large portion of the diversity. Here they are on Spirogyra, a colonial diatom, and even dinoflagellates. Many of these chytrids have never been cultured, and attempts have been made to try to culture them, so they're considered to be unculturable if one believes in that kind of thing. Here at University of Michigan, we are developing a resource which we call the Collection of Zoosporic Eufungi at the University of Michigan, or Cesium. And this is a resource that is built on two large legs. The one leg is that of Joyce Longcore, and the other leg is colleagues of mine from Alabama, Martha Powell and Peter Letcher, both of whom independently cultured a large number of these fungi and have contributed them to our collection in order that we preserve them and disseminate them and increase research on them. And these are all cryopreserved in liquid nitrogen. So using these cultures, we have been pursuing the phylogenetic systematics of the group. And this slide has got a lot of detail, but the important thing is that we have about 140 taxa, and we've sequenced their genomes, and we found we're using about 758 genes, proteins, to create a phylogeny. I guess we've filtered out some taxa and some proteins as not being very useful. And then we've used both concatenation and coalescence approaches to make one single phylogenetic hypothesis. And that looks like this, which is obviously a very difficult to read slide, but the point that I would make would be that there is a large group of fungi, the Chytridiomycota, that contains numerous lineages that we've now sampled. We have kind of mapped on a bunch of characters, including like whether or not they have a flagellum, what they have in terms of cell cycle regulatory proteins, the use of the amino acids selenocysteine, and cell wall proteins as well. And all the divergences in this group between these major phyla happen in the Precambrian, so long time ago, and yet they still persist today. And just to map on one of the characters, because I know otherwise there's not a lot that you can see of detail. Selenocysteine usage is really spotty in the tree, but it's found in many of the major branches. And is an example of a character that, it's clearly homologous in all of these lineages, which is what's interesting. But it's the same proteins that are involved. It's the same tRNA. It doesn't look like something that was horizontally transferred, but it's just something that almost all of the fungi have moved away from today. So it's interesting to think why that relic remains in some taxa and what it was about previous times where that usage of this amino acid was more favorable. Okay, so now I've talked about the chytrid phylogeny and introduced some of the major groups. I want to talk about what we're doing with this dark taxa and how we're trying to understand better what it's composed of using single-cell genomic approaches. Okay, so starting with two example phylogenies from the literature. This paper is a really important paper in zoosporic fungal biology because it proposed two orders. And these orders were described on the basis of material that had been brought into dual culture. So they were algae that were infected with chytrids. They were not brought into pure culture. But in being able to put pictures and names on some sequences, they were able to really look at the environmental data that's in GenBank. So all the stuff that kind of is not in bold and has weird names, those are all the environmental sequences. And you can see that there are many of them in this chytrid part of the tree. So this is all that I'm showing here, chytrids. And in a lot of cases, they're not related to any named taxa. So this whole order, Mesochytriales, is based around this one observation, but it's present in many different ecosystems, likely algal parasites. And then this paper introduces a branch in fungi. Actually, it's two branches that are at the base of the very kingdom. So here's the outgroup here, and here's sort of most of the described phyla of fungi. And then these are two branches that are related to the rest of the fungi. They're called Freshol or Freshol1. Okay, so there is some true dark matter out there, and our goal for our lab is to try to understand what these things might look like and what they might be doing in ecosystems. So what do we know about these dark lineages? Well, we know that they don't generally match the things that we have in pure culture. And we do know, generally speaking, where they go on the tree. And we know where they were collected from. But what we don't know is what they're doing in nature, because we generally only have the DNA sequence only. So we also don't know their morphology, and we don't know their enzymatic potential. And so the next parts that I'm going to talk about are largely a collaboration with my former postdoc, Kensuke Sato, who is considered, in my perspective, a real Jedi of chytrid biology, and someone who has an amazing grasp of the natural history, the systematics, and taxonomy of the group, and is a truly inspiring researcher to work with. In order to, I guess I should stop here and say, in order to link these environmental DNA barcodes to something that's happening in nature, we wanted to use a single cell approach. And that single cell, we wanted to be able to observe it. Here I'm going to show like the typical single cell workflow that you have in terms of getting genome sequences from individual cells. You have a sample, and then you have to isolate individual cells, and there are many ways of doing it. So laser capture microdissection, flow cytometry, you get single cells, and then those undergo a DNA extraction or just a simple lysis procedure, And then you go into this multiple displacement amplification, or essentially a whole genome amplification step. In our lab, we've taken two approaches from there. First, we'll do a long PCR and amplify a large region of the ribosomal RNA, and then sequence that in order to see what the phylogenetic placement of that strain is. Then if those strains are interesting, we'll take that same MDA product, run it into a sequencing library, and then do whole genome sequence of that. So more specific to our protocol, we would take pond sample or a soil sample, and then we look at it in the microscope. And if we see something that looks chytrid-like, an infected cell, for example, we'll isolate that cell, photograph it, and then undergo the whole genome amplification, PCR, and sequence the barcode, and then ultimately genome sequence. And here's a picture of Kensuke working hard at the inverted microscope, using just a simple glass pipette to isolate individual cells and wash them. And so, by serially transfer them across drops of water, we get rid of most of the contaminants, then pick up that final single cell and then put it in a tube. So Kensuke was able to, over about 2.5 years, he tried to isolate about 300 single cells that were all photographed, and he was able to get our DNA sequences for over 100 of them, and they represent 50 different lineages. And this is the taxonomic distribution of them, from Freschel L1 up to Blastocladiomycota. In this tree, the blue are the single cells, and then red are metabarcoding, and black are the reference taxa. Obviously, you can't see any of the detail, but just to give you an overview of what that looks like. Getting in on some of the detail, and this is from slightly earlier work that was done by two postdocs in the lab, William Davis and Alicia Quant. Here we are at Smith's Fen, and we observe this chytrid on this alga, and this is Desmidium, and you can see, these little circles here are the actual chytrid parasites, and these were put through our single cell sequencing pipeline. This is just with an 18S sequence, and compared that to things that were sequenced using the metabarcoding approach. And then you can see that we had a really good match with this parasite and something that was collected from the same fen. So now we're able to put some information onto this OTU that previously was just an OTU. Now we're able to say, okay, this looks to be an algal parasite, and the host range is at least Desmidium, and it looks to be epibiotic, and so that we're at least getting some way of both, saying what this thing does in the ecosystem, and also now we could potentially go back and find it again. This is an important thing. And this is something we are also trying to work on, is establishing dual cultures, although this is pretty tedious. Assuming this thing can't be brought into pure culture, which we also do try to do, we can try to grow the Desmidium and recombine it with the parasite to make sure to keep those things going in the lab. There's only so many of those you can do before you spend all your time trying to keep things going in the lab. One of the things we have kept going in the lab is this really interesting winter diatom parasite. So this is a collaboration with Mike McKay's lab at the University of Windsor. And here's Lake Erie. The ice is starting to thaw and break. And this brown stuff under the ice is all these diatoms, which seem to thrive in this habitat. And of course, where you have algae, you have chytrids. And here are some photos, many photos of individual cells that Kensuke sequenced that are from these habitats. And there's a couple different kinds of algae. This centric one, and then this colonial one here. And the OTUs all kind of fall in these 2 or 3 OTUs that are in this part of the tree in the Rhizophydiales, and they do associate with other environmental sequences that we have. So now we have a pretty good handle that there is a clade of chytrids that is specialized on these winter diatoms. And this slide is just showing how we are starting to go from those individual observations to now trying to put those single cells onto a tree. And I'll go ahead and talk about, sorry, the slide got slightly out of order as I rearranged it at the last minute. Some of these, I'll go ahead and explain what this is. So this is collaboration with the Joint Genome Institute, and we had cells that were separated out by flow cytometry. So here were cells that we didn't actually see with our eyes, but we sorted them out and we sequenced their genomes and we put them in the tree. This sigma is a measure of genome completeness. So from one single cell, how much of the genome can you actually get? It can range. So we're going to have as low as 15% of a genome to maybe 50% of a genome with that just single cell approach. And those can be readily placed in a tree. And as soon as we can do that, we can start to infer something about their biology. And with poor genome representations, you do have this issue of you can't really say what you don't have. If a gene's not present in a genome, that may not tell you anything, but it can tell you about the genes that you do, or the genes that you do have can tell you something about the function of those organisms. So this is just one example with a particular enzyme, thymidine kinase, which is involved in basically utilizing DNA that's taken from the host to and be incorporated into parasite DNA. And that's some salvage pathway that's used by microsporidia parasites and presumably also these other Cryptomycota, Rosella mycota parasites. And so the point I'm trying to make here is that we see the presence of this thymidine kinase in this single cell genome from Smith's fen. And so we can imagine that, this also goes in with this absence of ability to synthesize the building blocks of DNA. It looks like this parasite is probably stealing DNA from its host to use and to build its own genome. So that little story was about the samples that we did with flow cytometry. But now back to the samples that we did with microscopy paired with whole genome amplification and then genome sequencing. And these are the ones where we had the pictures and the Kensuke cells. And we've done a pilot now with about 10 cells, and these are the pictures of these cells. So you can see they're not just a cell. I mean, here's a diatom that's infected by this chytrid. Here's the diatom here. We had done the barcoding and we knew that it was placed into Rhizophydiales, which is a chytrid order. And then it also had a parasite. So somewhere inside here is a parasite of this chytrid, a hyperparasite. And then you can see some of the cells are complicated. So we have a rotifer here, and others are simple. Like this is a broken off sporangia is one of our targets. So one of the issues was as we get these samples that have a high complexity, so there's host DNA in there, presumably, and then there's bacterial DNA in there for sure. We have to be able to separate out the wheat from the chaff, so to speak. We need to be able to focus in on the genome of interest. And so we've developed an approach we call SQUID, or single cell genome ID, which assumes that the data coming in are likely to be metagenomic in its nature. And there are some differences in metagenomic data and single-cell genomic data having to do with the whole genome amplification. But our approach takes 3 different methods, one being the GC content of a particular contig in the genome assembly and the coverage of that contig, so how many times it was sequenced over and over, is one of the methods. And then we also take codon usage, so which codons does it use out of the synonymous ones, so that kind of bias can tell you what genome this contig might have come from. And then there's the actual underlying frequencies of the nucleotides when you consider 4 of them at a time. So runs of 4 nucleotides called tetramers. And those are diagnostic for species. So just to say that it's somewhat complicated. You assemble into this metagenome, and then you have to find a way to separate out your target. And here's an example of a very challenging target. This 142, this is presumably, I think it was a nematode egg, is what we assumed it was. And here's a topology map using those tetramer frequencies. And overlaid on top of it are blast matches. So in yellow are the bacterial and archaeal matches. And then these other regions, and so I should say this is a self-organizing map. So based on those tetramer frequencies, they kind of move into space, and things that are similar tend to go together. And you can see this patchiness in the blast match and the tetramer frequency. And the parts that we isolated out as the fungus of interest is shown here in orange. And yeah, I won't really go into this GC plot, but you can see that it's very complicated. There's a lot of different GC values for the different contigs. A table, it's a little bit just numbers here, but there are interesting details here. So this is the number of contigs that we got that we're considering to be, or this is before filtering. And then this is the size of that metagenomic-like assembly. And then after we filtered the assembly to just retain the fungi, these are the numbers of contigs that remain and the genome sizes. So some of them are quite small, and then some of them are, about normal fungal size. Then this last column gives us an idea of how complete the genome is based on presence of conserved eukaryotic orthologs. And some of them are quite good. We have maybe, almost the whole genome present. And others, range down to quite poor, actually. But with that amount of data, we can start to infer their biology and certainly infer their phylogenetic placement. And this is just a quick tree showing how we're able to start discovering some really interesting new lineages here. So all the ones in red are the cells that we sequenced. There's this lineage that's related to Blastocladiomycota, this Opidium-like thing in a rotifer egg. Actually, this must have been a rotifer egg and not a nematode egg. And then we have a few things scattered around the tree. And here's our sequence of the freshole, which we find to be at the very base of the fungal tree. Now, in summary, we've shown now in principle that we can start to go into the environment and take things that have never been brought into culture and to start to put information into what that dark taxa might be doing and what it might look like. So, once we get a genome, then we can start to do some of the biology or infer some of the biology. And I didn't talk about it, but we've done a lot of work to look at the ploidy that you can get from a single cell. And that tells us about whether it's diploid, haploid, or aneuploid. it's pretty easy to tell if the organism is flagellated because flagellum is a complex structure that requires many genes. We can look at the mitochondrial genome and see what genes that encodes, whether there even is a mitochondrion, and then we can look at nutrient transporters to think what it might be acquiring from the environment. We can start to look at defense genes, start to look at the cell wall, what it might look like. And then there are some markers for hyphal growth that we can also use. So we can start to see, well, we didn't see it make a hypha, but maybe it can make a hypha. And of course, carbon utilization genes important in terms of thinking about ecological roles. Oh, and even endosymbionts. So one of the things that comes out of these single cell analyses is that there may be things actually inside the cells, bacteria, viruses, and those are also interesting to understand the organism in a holistic way. So I've kind of gone through some of the various approaches to biodiversity discovery, highlighting the single cell genomes. It's not possible for us to move forward as a community thinking in only one way and ignoring another. So we do need to use both culturing approaches, metagenomic approaches, and single cell approaches. Metagenomic approaches are high throughput. We need them in order to be able to look at communities quickly. And they're very simple to do. Cultures are required fundamentally so we can do experiments, and ultimately phenotypes are much more straightforward when we have cultures. Single cell genomes give us this taxon sampling that's inaccessible by cultural methods, and it allows us to look at all of the genes in a cell, which is something you can't do with a metagenomic approach. We can also get these associated taxa. Could be bacteria in and on the cells. And at the center of it all, we're trying to get at the function of these fungi in nature, which is one of the major unknowns or major areas where I think we need to work as a community in an integrative approach to collaborate on understanding what the role of these fungi are in the environment. The dark matter fungi especially. So the take-home points is that there is a large percentage of fungi which are essentially dark matter because we don't know what they look like, and we don't know what they do in the environment. The zoosporic fungi have diversified, since the Precambrian and are represented by many different lineages, each of which is equally fascinating in terms of their basic biology. Many of them are uncultured and many of them are parasitic. They do important things like attack algae. And I went through how single cell genomics can change how we can start to break down these dark matter fungi to start to put some, at least initial glimpses into what these fungi look like and what they might be doing, with the ultimate goal of all of this to be able to take a genome sequence and predict the form and function of that organism that has that genome. I want to thank the organizers again for this opportunity to speak to you today. I have many people to thank, particularly Kensuke Sato, Kevin Ames, and Rayburn Simmons for their contributions to Chytrid systematics and single cell genomics. And our partners in single cell genomics have been colleagues at the Joint Genome Institute, also Mike McKay. Thanks especially to Stephen Arndt and Igor Grigoriev. And I think that's where I'd like to end it. I want to thank you all for this opportunity again, and I'd be happy to take any questions.
Thank you very much, Tim, for your interesting talk. We will go to the question and answer. Bueno, para abordar la sesión de preguntas y respuestas, en esta ocasión vamos a incluir a una alumna del posgrado en Ciencias Biológicas, Melissa Escárpita, para que participe y modere la sesión de preguntas. Adelante, Melissa.
Gracias, doctora. Thank you, Dr. Tim James, for this fantastic talk. Now we start with the Q&A session. We already have some questions. Martin Ricker asked, the large undescribed fungal diversity is often described nowadays genetically with operational taxonomic units. Aren't those OTUs rather disconnected from true formally described species, or the other way around? What is a fungal species?
Okay, starting with the hard questions. Wow, I think OTUs have a place in fungal ecology. They're placeholders, and other people use different names like species hypotheses. I think that's the thing that the Unite folks like to use. They certainly don't equate to species exactly, and species can't really be, in my opinion, can't really be fully described unless some biologist is actually going to take the time to figure out what's a species and what isn't. So I think that does create a bit of a gap, but I would say that in some senses, like, for example, I gave an example of a Syncytium OTU. In this particular study, we sequenced with PacBio. We made OTUs. There was a thing we called an OTU that had a sequence that was distinct. And that later matched something that we found in a single cell. And when you look at it in a phylogenetic context, that OTU is represented or is a representative of that species. So by tracing that OTU, we're kind of following a species, but not really. So there are a lot of caveats there. The broader question of what defines a species is a really interesting question. I think I sort of ascribe to the evolutionary species hypothesis, having to do with genetic isolation of a particular population. And none of that I can address with this particular study. But, when we start to look at, how many species there are, if we look at Hawksworth estimates of maybe a couple million species, and then we use some OTU approaches, as crude as they are, we get to a similar ballpark. So I admit that there's a lot of error in using the OTU terminology, but there's a strong correlation too. This is a good question. Thank you.
Thank you. Ricardo asks, do you think that it is wrong to use these unknown OTUs for the bioinformatics analysis when there is still a lot of knowledge about them and the analysis can be overestimated?
Well, it depends on which analysis we're talking about. So as far as I understand it, like in the microbial ecology community, they're even moving to, they understand that they don't equate to species, these OTUs. So they're moving away from OTUs and going into sequence variants. And both OTUs and then like sequence variants, which is just every unique sequence is treated in a different way, are both proxies for what we're really after, which is an underlying species diversity and composition. And so in my particular work, we use phylogenetic approaches to try to get a handle on what might be a real species. Without being able to do any of the experimentation, it's pretty loose. So, we look for clusters in our trees. We can't do any crosses. We have a rough handle on phenotype. So I don't know if this question has anything to do with the overall estimate of total fungal diversity, but those original estimates by Hawksworth really were not dependent on any. I mean, this was in 1991 before all this crazy sequencing took off, and that was based on named species, which also have their limitations as well. There's a lot of synonymy and other issues that taxonomists have to deal with. So, thanks.
Dr. Maria Gonzalez asking, do you consider that morphology is still important to describe new fungal taxa?
I mean, I think as a working, systematist, I 100% think that it is important and critical, because I don't want to produce any kind of guide to taxa that's based solely on DNA. Nobody can work with that. Nobody can actually name a species if it's only based on DNA. That said, there's one issue with chytrids. They are very difficult, and even the masters, Joyce Longcore and Martha Powell, they have a hard time putting names on species, even things that they've been culturing for 40, 50 years, and they still have a hard time putting a name on things. When we use a phylogenetic tree, then you can start putting names on things. And it's a combination of both morphology and sequence that is needed in some of these more difficult taxa.
Julieta Álvarez Manjarres, amazing talk. Are there any differences between epibiotic and endobiotic parasitism in their genomes?
That's a good question. So both, I would have maybe a while ago said that, the epibiotic are sort of closer to free living, and the endobiotic are more like obligate parasites. Certainly because they're closer to the action in a way. They spend the endobiotics spend their entire existence inside the host. And I will say that those endobiotic parasites that are in the chytrids, Rosella and Opidium, those cannot be cultured, and their genomes appear to be quite reduced. But the epibiotic parasites also have some of these unculturable lineages like Zygorhizidium. And there they also seem to have pretty minimal genomes, and Kensuke is working on those. So things like 14 megabase genomes, which isn't too shabby. I mean, it's similar to yeast. But I do think that in general, to answer the question, that the endobiotic parasites are on average more reduced in terms of their metabolic potential and ability to live independently of the host than the epibiotic parasites.
Thank you, Doctor. Edmundo González Santillán, can someone of the biological interactions, endo, epi, parasitic, symbiotic, commensalist, can be used as diagnostic characters for some species?
We don't know too much about the specificity. We know that there are some facultative parasites, things that we can see in some cases, things that are on pollen can also go on to algae. I think that the endo and epibiotic is something that we can actually use. So that's a nice morphological characteristic that does show some consistency. The parasitic part is more of a continuum, although there are certain lineages that are really hardwired, just like the endobiotic ones I was talking about. They're so obligate with their hosts that those are actually tracking their hosts, and so we can use those as characters, and we believe that some of them show high specificity to their host. And yeah, so and then the commensalism is an interesting thing that we know very little about in chytrids.
And Omar Lagunas, how do you know that chytrids are parasites of algae? Could they live as epibiotic or endobiotic and not as parasites?
Yeah, It's a good question. We can do a test, and it's about as simple as you can imagine. So we can take the algae and grow them in a tube, and then we can add the chytrid to it. And then does it turn brown? Does it turn clear? And that we will call as a parasite. Now there's a one thing that Rhizodium and Phycophyllum, which is considered to be a mutualist of algae. And when you grow them in co-culture, they promote each other's growth. But generally speaking, when we see these chytrids on algae, they'll stick a rhizoid into the host, and that's likely to be where they're drawing their nutrition. And you can even see some cells that, if it's on a filament, they're really attacking that specific cell and drawing out the nutrients from it. And they're well known to cause crashes. One of the things that we're trying to help with is that when people are trying to grow algae for biofuels and they cultivate them outdoors, which is what you want to do because you can use natural light and everything, chytrids come and they often crash your cultures.
Thank you, Doctor. And Dr. Ulises is asking, I noticed that you use nanopore tech for sequencing whole genomes of your single-cell isolates. Any particular reason for choosing nanopore as opposed to PacBio? Great talk.
Okay, well, thank you so much. There was a method proposed for long amplicon barcoding. So this wasn't genome sequencing, but actually the PCR sequencing. And it was mostly because a method had been developed and you can do it in your own lab. We used to have PacBio on campus and it kind of went away. So the nanopore is really fun. And, we also published this paper on the chytrid collection, the culture collection. And we made a lot of progress with that culture collection by barcoding with nanopore. And, I would actually consider that people should consider doing that for their other fungi because the cost was really cheap and the accuracy, we figured it was more than like 99.5% accurate compared to Sanger sequencing, which is really accurate. And we could get like 6,000 base pairs of data, and all that's generated in your own lab.
Thank you, Doctor. And I have a question. Now that you have these genomes and have a lot of information about the metabolism of these species, is it possible to design a culture medium that is capable of isolating these fungi?
That is that's kind of $1 million question right there. And it's definitely the kind of pie in the sky stuff that we've been wanting to do. And it's doable to some degree, yeah, but it is also a major challenge too. So on one end, we're going to have things that probably no, they just require too many things, or they sort of have a mechanical or some really complex relationship with the host that we just can't get there. And then there are going to be things on the other extreme where it's like all of a sudden we see, oh, all it needs is a vitamin like biotin because it can't make that. Add biotin, all of a sudden we're going to grow it. And then there's all the stuff in the middle, which is going to require probably some experts in biochemistry to help point us to the things we should be looking for. But again, that's a great question and something I'd like to do.
Thank you, Doctor. Well, you have a lot of congratulations for your amazing talk. Dr. Patricia Velez say thanks for the excellent talk. Looking forward for Kensuke paper. Thanks a lot, Dr. James, for your excellent presentation and really interesting seminar. Amazing talk. You have a lot of congratulations. Thank you so much for being here with us and share your knowledge.
It's a true honor. Thank you.
Thank you very much for your interesting talk. And thank you, Melissa, for support with the answer and question session. Well done. And we will go with Ulysses for close this symposium.
Well, we want to thank Professor James for giving this fantastic talk. Muchas gracias, María del Carmen, por ser la anfitriona de esta sesión y Melissa Escárpita por haber apoyado con las preguntas y respuestas. Con esto, pues damos por terminada la sesión del día de hoy. A nombre del comité organizador de los Seminarios de Fronteras, les recordamos que la próxima sesión va a ser el próximo martes 29 de marzo a las 11 de la mañana en este mismo canal. Y también les recordamos a la comunidad del Instituto de Biología, particularmente los académicos y estudiantes, que el día de hoy vamos a enviar la encuesta para las personas interesadas en platicar con la profesora Kelly Zamudio de la University of Texas at Austin, quien nos va a hablar de Global Impacts of the Amphibian Killing Fungus, a Functional Genomic View. Estas reuniones post-seminario han sido muy importantes y, pues, por ejemplo, el día de hoy también tendremos una con el profesor Timothy James, con algunos estudiantes y académicos de nuestra institución. Pues muchas gracias y les damos las gracias al doctor Timothy James y pues nos vemos la próxima sesión. Gracias a todos.
Muchas gracias.